Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. We are covering a course Python for biological data analysis. Today we are going to cover chapter 5, that is NumPy for numerical data or NumPy for numerical data analysis. So let's start. Okay, first of all, I'm going to open my Chrome browser, and this is Chrome browser. Now you have to type Colab or simply use Google Colab. Now you will see this kind of uh, window. Click here. Now click GitHub. In GitHub, you need to mention ASH in the search box 100. ASH 100 and tap enter. Now click here and scroll down to Python for life sciences. Life sciences. Now you have these. I will remove one of them, but you have this chapter 5, NumPy for numerical data. Both of them are the same. I will remove one of them. That is okay. So you have to click the NumPy chapter 5. Click here. Now click connect here. The notebook will be connected. Now you are seeing the course name. Now chapter 5 NumPy for numerical data, and the course is designed by me. My introduction. And during teaching, I will use examples from the biological sciences or life sciences. So once again, I want to emphasize that if you are from not from the biology background, if you are from computer science or related field, then I'm afraid this course is not for you. Until now, we have covered chapter 1, Chapter 2 Control Structure, Chapter 3 Data Structure for Biology, Chapter 4 Working with Files. If you want to watch these chapters, then simply you can click these links and it will lead you to the particular video. Now, today we are going to cover NumPy for numerical data analysis or we in this lecture we will get familiar with the NumPy, what are arrays. So let's start. NumPy is actually a foundational library. Uh, when we talk about Python, so Python is a kind of you I would say it is a kind of sea, river, ocean. I mean, it's a very huge platform. So if you are interested in data analysis, then most of the people or majority of the people they are using NumPy. It is a foundational library. You can say like if NumPy is a room, sorry, if Python is a room full of books, of different books, then NumPy is one of their book placed in Python. This is the simplest explanation I can give it to you. It's very easy and very, very powerful. So NumPy is a foundation library in Python and we are using it for numerical computing. In particular, for the dataset that contains numbers, numerical computing. So in biology, we often deal with large dataset, for example, gene expression levels, protein concentrations, and so on. So we will also deal some example in this course. So NumPy will provide you the easiest and the most powerful way to deal with such kind of data. What is NumPy array? NumPy array is actually a grid of values, all of the same types. All of the same type means all are numbers. Okay, and they are indexed by a tuple. What is actually a tuple? We have already covered it, I guess, in lecture 3. So, you can think of a library which is so much powerful to comprehend, to take care of all the numbers in the datasets, all the numerics in the dataset. So NumPy is the solution. So when we are working with a NumPy, so we will use this statement: import NumPy is MP, NP. So NP stands for NumPy. So what it does, if we are using Python, but we ask Python to, I need to use a book named NumPy. I need to use a library named NumPy. So once you incorporate or once you run this statement in the start of your analysis, then that your work session or workspace will automatically be used to all those functions. Okay, let's create an array from a list of a number. For example, these are some numbers: 2.5, comma, space 3.1, comma, and so on. So I name it measurements is equal to what is numpy np dot array. It is a function which is built in the NumPy. So we already imported NumPy as NP, so that's why np.array will work and the program will recognize it. So measurements is equal to np.array, and then put your data in this format, smaller brackets or parentheses, or followed by a larger one. Okay, then print command, and simply use this name here for the print command. It will be printed. Okay, now you can say the whole data is printed here. So now this is the easiest way to convert your numerical data into NumPy array. Okay, in this context of biology, this array could represent the length of bacterial cells, or micro in micrometer, or the optical density reading of a culture over time, or some other thing, for example protein concentration or some gene expression data, whatever the data is. Okay, now, how we can perform some mathematical operation easily uh, on the data, which is we read it in the form of NumPy array. For example, we have a data, kind of just general data. So let's suppose this is our data, and the data is in micrometer. So I name it micrometer, let's say, and I'm interested to convert the whole data into nanometer. Okay, so first we will create a data frame. So we also call it a df. So micrometer is equal to the same thing, num np dot array. So we just created a numpy array. Now we are going to convert this data into a nanometer. So when we convert something, a micrometer to a nanometer, so we need, it's simple mathematics, we need to multiply micrometer with 1000. So we are doing the same. So now we are creating another data frame. Nanometer is equal to micrometers multiply by 1000, and then we use the print command. So you can see measurements in nanometer, uh, 25 instead of 2.5. they give me 2500, and they give me 3100, and so on. So, do you see? Simply, you convert your data into the NumPy array using the NP array, and doesn't matter how much longer that data is or how much complex that data is, until and unless if it is a numerical data. Now, we are going to perform some operations. So let's say we have two different sets of array. For example, this is set one. You can say, let's say you are a student of biology. So, for example, this is gene expression data of one sample, and this is the gene expression data of another sample. So I name it set one np.array some data, and then set2 np.array again some data. I am interested to calculate the difference between them, set1 minus set2. So the difference is equal to set1 minus set2, and then print it. You can see that 2.5 minus 2.6 is minus 0.1, and etc etc. Now, we can also perform some statistical calculation. For example, we have some data. Let's say we have heart rate or heartbeat rate, the measurement of heartbeat rate. So the data is equal to np.array. First, I convert that data into the NumPy array, and then now I'm interested to calculate the mean. So what should I do? I will create another data set, average heart rate. It's up to you what name you choose it, and np.mean. First, when we were interested to convert the data into array, we use np.array. Now I'm using another function np.mean. Okay, and then this data is actually this name, this data frame you created here. If you created like if you mention here one, then you not one. It should be some name. Let's say if you created Ash, then you should mention Ash here. So it will calculate the mean. So the mean is 123. Okay, now the same way I'm interested a median for some data. This first the gene expression data, let's suppose I converted to the NumPy array. Then here I generate another data frame, and it will be np.median gene expression, and the median is 12. Okay, if you have attended my previous lecture, I told you like things are getting interesting in future lectures. If you remember, in my lecture 2 and 3, I discussed definition and loops with you guys. So in this exercise or in this chapter, when you are dealing with numerical data, so you have two options, either to define and use those for loop or while loop, and it will make things more complex. But if you are using NP or NumPy, so you can see without definition, we can calculate, we can sort out different things very quickly, and it do not also it do not need much more expertise, and you can easily uh, uh, equip with these kind of skills. You can easily equip yourself. Okay, now I'm interested to calculate standard deviation of some data, let's say the heartbeat rate data again. So the data NumPy array I made a NumPy array. Now I'm interested to calculate the standard deviation. So I'm using np.std this time. And this is a new data set. I name it. Just click here. So the standard deviation is 4.1. Now let's explore these things with some experimental measurements. Suppose we measure enzyme activity in micromole per minute across 5 samples. So the enzyme activity data I have is np.era. First, I'm converting the enzyme activity data into NumPy array and then print it. So you can see that was just a print command. I did it. Now I'm interested to normalize the whole data. So what I do, mm, I'm interested to not only to normalize to find out the mean enzyme activity. Also, I'm interested to normalize the whole data. So if you see, the mean activity data frame will be equal to np dot mean, and here will be the name of our data frame, which explains our data or which records our data. So enzyme underscore activity, you can see here, this data frame is already stored with us. So we will use np dot mean enzyme underscore activity. Wha- if we are going to change this name, let's say I'm going to change this name ABC, just to show you like it will throw an error. Okay, now I also am going to change here ABC. So it's throwing an error. Name ABC is not defined because the program does not know like where it, what is ABC. So, but if I do this thing, okay, if I change this thing to ABC, this error will be gone. But once again, you copy this and paste it here, and instead of ABC, also here. Okay, what is the normalized activity? So the in normalization, we normally are doing enzyme activity divide by the mean activity. So that is called normalized data. So now normalized data is 0.8, 1 point this. Okay, so this was kind of enzyme activity normalization. Now we are going to make some, uh, we are going a little further. For example, we are interested in a fold change in gene expression data. So for example, we have the control expression data. So first we will convert it to a NumPy array 100, 200, and 150. Suppose we have a treated samples, also convert them into 304 and 225 into NP array. The same thing we did it with set 1 and set 2 here. Now we are interested in a fold chain. So the fold chain will be treated data divided by the control experiment. Okay, like here, these are the names. So you can see. So in first case, we have a fold change, three-fold, two-fold, and 1.54. So you can see very easily we just put our formula because in NumPy we are allowed to do subtraction, addition, mean, mode, standard deviation, whatever you are interested, you can do it. Now we are interested to calculate the mean, median, and standard deviation uh, in that enzyme activity data. So here we just ask to print mean, and what to print? np dot mean. This is a function. Mean of enzyme activity, median of enzyme activity, standard np dot std of enzyme activity. You run it, you will have all these three data easily. Now, I put some exercise for you. This is a protein concentration data, np.array. You need to first run this. So this data will be recorded. Protein dot conc stands for concentration. Now, your job is to calculate mean, median, standard deviation, and normalize this data set, these four things. I put four empty cells for you. If you are interested, you can generate another one also, just clicking here the code and just try yourself. This is your data frame, and now calculate these things. Let's see if you can do it or not. So that this was all for this chapter, like how what is NumPy and how we can use it for different mathematical calculations and for experimental data and all other things. I hope you like this class or you like this lecture. So stay with me for more interesting lecture. Thank you so much. Bye bye.